Good morning, everyone. Like Ms. Polisi said, I've been working with Dr. Lexi at Brooklyn College, and our main focus is on cell adhesion, and we only work with yeast cells. And this is an outline of what we will be going through today. We will start from the introduction, and we'll go through material methodology and all the way down to acknowledgments. So we'll start off by discussing a little bit about what cell adhesion is. Well, cell adhesion is the binding of a cell to a surface, such as an extracellular matrix or another cell using cell adhesion molecules. Well, one protein that we have been working with is the ALS5. It is a protein which is actually responsible for cell adhesion in most of these cells. And what we perform is a cell aggregation assay to see the binding of the cells. So the particular type of yeast cell that we work on is Candida albicans. And Candida albicans is from the kingdom of fungi, and they are the most common cause of opportunistic fungal infection worldwide. And some of the latter outcomes of Candida infections from Candida albicans are some people that suffer through them, or people already suffering from HIV, AIDS, and other transplant patients. So in my hypotheses, Candida albicans will form huge aggregates if a cell aggregation experiment is done binding it to BSA-coated beads. So BSA-coated beads are magnetic beads which will attract the SC53 strain of Candida albicans, which I've been working on, and large aggregates will verify that the experiment was conducted properly. So these are some of the materials that are used throughout the experiment. Some of these materials include YPD media, OD spectrometer. OD spectrometer is a really important technology which we use in calculating the amount of cells after they grow in a culture, which I will discuss later in the methods how that is done. And the way they calculate the amount of cells in a test tube is by when we place a test tube in this OD spectrometer, there is a light that passes through the test tube horizontally, which is proportional to the number of cells in the test tube. So in my methods, we make a YEPD plate by pouring the media into standard petri dishes. We allow the YEPD media to solidify in the petri dish for around 2 days. We streak the SC5314 strain of Candida albicans on the YEPD plate and leave the plate in a 30 degrees Celsius incubator. So when we leave the cells in, is that a question?
I just want to ask, what is the YEPD stand for?
Well, YEPD, it really doesn't stand for nothing. I mean, it stands for something, but in the material, you can tell these are the ingredients that are used, but in general, what YEPD plates are used for are to create single colonies, which later we inoculate into YEPD media. And overnight, when we put these in the single colony with the YEPD media into something known as a lab shaker, they grow overnight into a culture of cells. And then, as I said before, we measure the amount of cells using the OD spec, and the volume of SC5314 and YPD media to pour into each test tube is calculated. After pouring a total of 3 millimeters into 4 test tubes, we centrifuge the test tubes in order to isolate the cells from the media. So how we know we need 3 millimeters is after we measure the OD, we know the amount of cells we pour, SC5314 we pour into each tube, and the rest we pour in is YPD media to make sure we get around 3 milliliters. And the reason why I use 4 test tubes is in case something goes wrong with one test tube, let's say the test tube breaks, or something goes wrong with how much YPD media or how many cells I pour into each test tube that we can use on the other 2-3 test tubes. So we add TE buffer after remove the media. The role of the TE buffer is to wash the cells. And after we put in the TE buffer, we centrifuge the cells once again at 24 degrees Celsius and 4,000 RPM for 3 to 4 minutes. So after the cells are washed, we remove the TE buffer and we add 1 million VSA coated dynamites. And the amount we add is 11 microliters, while 989 microliters were added of TE buffer. And as I said before, these dynamites, they attract the cell aggregates, which we later, when we put into the shaker and after they are activated, we put in the test tube in a magnetic holder for 2 minutes. And the magnetic holder, it attracts the dynamites along with the cell aggregates. And after that, after those 2 minutes, we remove the TE buffer from the test tubes while the cell aggregates are along the side of the test tube because the magnetic holder pulls them on that side of the test tube. So we remove the TE buffer, and then we add a TE buffer again, but we make sure that it doesn't interfere with the cell aggregates which are along the test tube. And when we remove the TE buffer once again, we make sure that the test tubes, the surface of the test, the bottom of the test tube is clear, and so there are no cells such as dead cells or any buffer. So lastly, we add 100 microliters of TE buffer, and then the aggregates are gently suspended and cells are viewed on a confocal microscope. And now I'm about to show some of the aggregates that were formed after I viewed them on the microscope. So as you can see, this is not a really that big of a large aggregate, but as I proceed, I'll show you large aggregates. And those really dark dots, those are dynamites. When performing this experiment, it is not possible to remove all the dynamites, but our goal is to remove most. And these are the 2 of the largest aggregates formed. So as a future consideration, the goal was to perform a basic cell creation assay, which I did, which will lead to more complex experiments in the future. And in the future, I would also like to perform an aggregation assay on Saccharomyces cerevisiae, and perform an experiment with ALS5, which I discussed before is the reason why cell adhesion is possible in most yeast cells. These are the references I used. Any questions?
Questions?
To wrap this presentation up, I would like to thank Dr. Licky and Dr. Ramsuk and Dr. Joseph, who are 2 of my mentors, the rest of Licky Lab, Dr. Sad, Ms. Khaleesi and Mr. Lee, the HSA staff, and all of you guys. Thank you.
I have a question. If you could take away one portion of what you learned, what is the one thing that's going to stand out the most for you?
Can you repeat that?
What is the one thing that you did, part of your internship, that will stand out the most?
Well, besides the research project, the honesty in the lab members, they were really honest. It was like a family,
Right.
and they were really helpful. I got to learn what each and every one of the lab members were doing, besides my lab mentor, and yeah, they really took their time explaining to me what was going on when I first walked in.
That always makes sense.
Okay, thank you very much. That was very informative, and I'm glad that you had a wonderful experience. I always like to ask, what is the one thing that you took away from your experience? Because that's the thing, that's what you'll remember the most in 5 years from now. Okay, actually what we're going to do, I had a few latecomers that I've seen walk in, so I'm going to give them the opportunity to come in, sign in, and you need to submit your July and August attendance sheet. So let's take a very quick 10-minute break before our next presentation.